Hello students, today we will study breeding methods for improving disease resistance in plants. Earlier we had studied structural and biochemical differences in plants. Plant breeding involves the plant breeders. Plant breeders emphasize the selection and development of disease resistant plants. Plant diseases can also be primarily controlled by use of pesticides and cultivation practices like crop rotation, planting density, disease free seeds, cleaning of equipment, etc. But plant varieties with inherent disease resistance are generally preferred. Breeding for disease resistance began when plants were first domesticated. Breeding efforts continue because pathogen populations are under selection pressure for increased virulence. New pathogens appear, evolving cultivation practices and changing climate can reduce resistance or is strengthen the pathogens, and plant breeding for other traits can disrupt prior resistance. A plant line with acceptable resistance against one pathogen may lack resistance against others. So this plant disease resistance is very important. Breeding for resistance typically includes identification of plants that may be less desirable in other ways but which carry a useful disease resistant trait, including wild plant lines that often express enhanced resistance. We can select wild plants for the disease resistance because they show enhanced resistance than the cultivated plants. Crossing of a desirable but disease susceptible variety to a plant that is a source of resistance. Here we had taken wild plant as a disease resistant source. We will cross this with the disease susceptible variety. Growth of breeding candidates in a disease conducive setting, possibly including pathogen inoculation. Attention must be paid to the specific pathogen isolates to address variability within a single pathogen species. Selection of disease resistant individual that retain other desirable traits such as yield, quality, and including other disease resistant traits. Resistance is termed durable if it continues to be effective over multiple years of widespread use as pathogen populations evolve. Vertical resistance is specific to certain races or strains of pathogen species. The resistance is of two types, vertical resistance and the horizontal resistance. If it is controlled by a single resistant gene, and it can be less durable, it is known as vertical resistance, whereas horizontal or broad spectrum resistance against an entire pathogen species is often only incompletely effective but more durable and is often controlled by many genes that segregate in breeding populations. Crops such as potato, apple, banana, sugarcane are often propagated by the vegetative reproduction to preserve highly desirable plant varieties, because for these species, outcrossing seriously disrupts the preferred traits. So, asexual propagation use here. Vegetatively propagated crops may be among the best targets for resistance improvement by the biotechnology method of plant transformation to manage genes that affect disease resistance. Thus, we had seen for sexual reproduction methods, the hybridization or breeding methods are most common, and whereas in vegetatively propagated plants, biotechnology is the main method for the transformation or the development of disease resistance plant. Scientific breeding for disease resistance originated with Sir Roland Biffen, who identified a single recessive gene for resistance to wheat yellow rust. Nearly every crop was then bred to include disease resistant genes, many by introgression from compatible wild relatives. Thus, we had seen how one gene resistant gene is incorporated in the whole wheat varieties to make it disease resistant. Some of the breeding methods are as follows conventional methods. It is an age old collection of methods which has been developed from ancient times, from beginning of human cultivation in society from till today. Various cultivars has been developed. The most renowned examples is development of semi-dwarf high-yielding cultivars of cereals during the Green Revolution and the hybrid rice developed in 1970s is here conventional approach has been used. Conventional methods involve Pureline method, which is developed especially for the self-pollinated crops. Since Pureline cultivars is progeny of single homozygous self-pollinated species, so they can be easily developed in nature and has importance in breeding of inbred lines that are used to make hybrids in self or cross pollinated crops, as well as they are desired in regions where uniformity of a product fetches a high premium. Though this method maximum possible improvement over the original varieties can be achieved and useful in improving low heritability traits since selection is based on progeny performance. Pureline method. Next is Pedigree method, in which the detailed record of the origin of the selected individuals or lines are maintained. Pedigree methods, we also maintain the records. Due to such records, we are able to know from which parent superior F2 progeny will develop. It is used for highly heritable traits like seed size, shape, disease, insect height, and maturity. It is widely used for the development of disease resistant varieties if governed by major genes. Variety developed through pedigree method is Lakshmi of cotton resistance for leaf blight, but the varieties developed through it cannot be used in environments where genetic variability for the characters of interest is not expressed. Requires a lot of time for testing along with intensive record keeping. Also, it is costly in terms of land and labor requirement. In case of characters governed by polygenes, it is not effective. Pedigree method is polygenic method or horizontal resistant method. Backrows. With the time, backrows variety has been developed by crossing F1 hybrid with any of the known parent and is useful in breeding for small grain crops. In this method, two plants are selected and crossed together. On regular backcrossing with one of the recurrent parent, the backcross progeny developed is almost identical to the recurrence recurrent one. In this method, the desirable variety, which are lacking in some characteristics known as recurrent or recipient parent, while the undesirable variety or wild variety has only one or two desirable characters. Interspecific hybrids. These are developed by crossing two species of the same genus with the objective of transferring one or two simply inherited characters like disease resistance to a crop species. Through introgression, genetic variation of one or both of the parents increases. Molecular approach. We had studied the classical approaches, now we will study molecular approach. With the discovery of molecular markers, this approach came into existence. The development of gene identification technologies using the tools of biotechnology provides ample opportunity for scientists to further improve modern cultivars, generally breeders use measured gene resistance for developing a new variety, but the passage of time it fails to provide the long-term disease control. It is rapid, but it fails to provide the long-term disease control. This is the shortcoming with the molecular approach. Multiline variety. It is the mixture of several similar pure line having different genes for disease resistance. Mixture of several similar pure lines having different genes for disease resistance. For development of multi-line varieties, several pure lines with different resistance genes should be developed through backrow programs using one recurrent parent, which is done by transferring disease resistant genes from several donor parents carrying different resistance genes to a single recurrent parent. Thus, we can produce multi-line variety. Marker assisted breeding. With the progress in molecular biotechnology, various types of molecular markers has been developed during 1980s and 1990s in crop plants. The rapid development of molecular markers, particularly DNA markers, and continuous improvement of molecular assays has led to the birth of marker assisted breeding MAB. Molecular markers have greater opportunity for improving the efficiency of conventional breeding by carrying out selection not directly on the trait of interest but also on linked markers of that particular trait. Next is transgenic strategies. It is an advanced form of genetic engineering in which foreign genes are introgressed into the genome of plant species for achieving desired goals. In this method, transgenic plants acquire a single desired trait above without any alteration of the original genetic makeup, which allows multiple gene insertions simultaneously into genome to obtain broad spectrum resistance lines. It is a novel strategy through which fungal diseases can be effectively managed by the introducing and overexpressing of genes that encode proteins which are involved in the synthesis of compounds toxic to fungi and with direct inhibitory effect on the growth of fungi. Thus, we had seen the genetic engineering or transgenic approach for the development of disease resistant plants. Next is the tissue culture approach. Tissue culture plays an important role in selection of explants. Explants which are selected either through toxins or callus culture. These toxins include phytotoxic metabolites which are generated by pathogens which may be from pathogenic fungi or bacteria. These pathogenic fungi belongs from Fusarium, Phytophthora, Alternaria, Helminthosporium, while a pathogenic bacteria is comes under the genera Clavibacter, Irvinia, Pseudomonas, and Xanthomonas. As reported by many researchers, they these phytotoxins are recognized as a useful tool for screening and breeding for disease resistant plants. Host susceptibility alleles. Most plant pathogens reprogram host gene expression patterns. Most plants pathogens reprogram host gene expression patterns to directly benefit the pathogen. Reprogrammed genes requires the pathogen survival and proliferation can be thought of as disease susceptibility genes. Recessive resistance genes are disease susceptibility candidates. For example, a mutation disabled Idiopsis gene encoding pectidlysis involved in cell wall degradation, conferring resistance to the powdery mildew pathogen. Similarly, the barley MLO gene and spontaneously muted pea and tomato MLO orthologs also confer powdery mildew resistance. LR34 is a gene that provides partial resistance to leaf and yellow rust and powdery mildew in wheat. LR34 encodes an adenosine triphosphate binding cassettes transporters. The dominant allele that provide disease resistance were recently found in cultivated wheat, not in wild strains. And like MLO, provide broad spectrum resistance in barley. Thus, host susceptibility alleles can be used for developing resistant varieties. Next is gene silencing. RNA silencing based resistance is a powerful tool for engineering resistant crops. The advantage of RNAi as a novel gene therapy against fungal, viral, and bacterial infection in plants lies in the fact that it regulates gene expression via mRNA degradation, translation, repression, and chromatin remodeling through small non coding RNAs. Mechanistically, the silencing process are guided by processing products of the double stranded RNA trigger, which are known as small interfering RNAs and microRNA. Thus, RNA silencing is a useful tool for the development of resistant crops. These are the some techniques or the methods by which we can get or we can make the disease resistant plants. In the next video or next class, we will study or I will give you a PDF or some how the how some disease resistant varieties of some commercially or agriculturally important plants has been developed in at plant breeding stations by the plant breeders, and we use them and they are showing disease resistant characters. Thank you.